Hello everyone, welcome to EPG Pathashala. I am Dr. Vinuta, Scientist at Division of Biochemistry, Indian Agricultural Research Institute, that is IARI, New Delhi, India. Today I am going to talk about enzyme inhibitions. Under this module, we will be going to explore on enzyme inhibition and enzyme catalysis. The need for studying enzyme inhibition is to understand in detail on various types of enzyme inhibitions. As you all know that an enzyme catalyzed reaction can be hindered or reduced by a number of substances, and those substances are called as inhibitors, given the conditions that these inhibitors must act in a fairly specific way in inhibiting the catalysis of particular enzyme, then only they are called as inhibitors. The objectives of this module is to explore on enzyme inhibition, to explore on the need for studying enzyme inhibitions, and to understand in detail on various types of enzyme inhibitions. An enzyme catalyzed reaction can be hindered or reduced by a number of substances. Some others like urea are known as denaturants, being non-specific in their mode of action. But if any compound act in a fairly specific way in inhibiting the catalysis of a particular enzyme, they are called as inhibitors. The loss in activity can either be of two types: reversible, where the activity can be restored by the removal of inhibiting compound, it is temporary; irreversible, where the loss of activity cannot be recovered within the stipulated time of interest. Heavy metal ions like mercury, lead, etc. cause irreversible inhibition, which binds strongly to the amino acid backbone, termed as suicide inhibition. Why we need to study enzyme inhibitions? There are several reasons behind the need for studying enzyme inhibitions, like exploring potential mechanisms in multi-substrate reactions, means how a particular enzyme behaves when different substrates are provided in a catalytic reaction. We need to study enzyme inhibitions for studying the relative binding affinity of competitive inhibitors to the active site in the absence of 3D structure information, and for understanding various control mechanisms, how the balance of protease enzymes and their inhibitors in tissues achieve homeostasis, and for various commercial applications like pesticide, insecticide, weed killers, pharmaceutical compounds like drugs, etc. For example, one should know the compound in pesticide, how exactly it kills pest, what are the specific enzymes it actually targeting without affecting the beneficial insects in the environment. In this type of inhibition, the hindrance or inhibition of enzyme is temporary, and thus non-covalent interactions like hydrogen bonds, ionic bonds or hydrophobic bonds form between inhibitors and the enzyme. Even though these are weak bonds, multiple such bonds causes strong and specific binding. Despite absence of any chemical reactions, the inhibitors can easily remove or exchange by dilution or dialysis. Types of reversible inhibition. Reversible inhibitors are of different types. They are classified according to the effect of varying the concentration of the enzymes, substrate and the inhibitor. Those are competitive inhibition, uncompetitive inhibition, non-competitive inhibition and mixed inhibition. Let us see the competitive inhibition. In this type of reversible inhibition, substrate and inhibitor cannot bind to the enzyme at the same time to the allosteric active site. This normally occurs due to the structural similarity of substrate and the inhibitor, which results with the affinity for the active site. The inhibition can be recovered by the presence of high concentration of substrate outcoming the competing inhibitor. Vmax of the reaction is unchanged, while the Kd, the dissociation constant, is apparently increased. Competitive inhibitors can also be used to find the active site of the enzyme. Clinical use of competitive inhibitors. There are several inhibitors which are available which can be used for clinical purposes. Uncompetitive inhibition. It is an anti-competitive inhibition where inhibitor binds only to the substrate enzyme complex. According to its kinetics, Vmax and Km decreases. This type of inhibition works best when substrate concentration is high. Uncompetitive inhibitor does not resemble with the substrate. Example, lithium and phosphoinositide cycle. Non-competitive inhibition. A non-competitive inhibitor reacts with enzyme substrate complex. It does not affect the binding of substrate. It slows the rate of reaction to form the enzyme product complex. The extent of hindrance or inhibition depends only on the concentration of the inhibitor. Vmax will decrease and Km will remain the same. Example, alanine non-competitively inhibits the enzyme pyruvate kinase. There are several common non-competitive inhibitors are given in the table. Mixed inhibition. In this type of inhibition, the inhibitor is capable of binding to both free enzyme as well as enzyme substrate complex. In this case, Vmax and Km varies. Mixed inhibitor binds to the allosteric site. This type of inhibition cannot overcome by increasing the substrate concentration, but can be reduced. The inhibitor binding to the allosteric site changes the structural conformation to reduce the affinity of the substrate. Irreversible inhibition. In this type of inhibition, the hindrance is of permanent nature by modifying enzyme covalently. These types of inhibitors often contain electrophilic functional groups like fluorophosphates, aldehydes, alloalkanes, alkenes, nitrogen musters, phenyl sulfonates, Michael acceptors, etc., which reacts with amino acid side chains having nucleophilic residues. These inhibitors are very specific in the mechanism of inactivation for a particular class of enzymes. They do irreversible inhibition by specially altering the active site. They display time dependent inhibition and their potency therefore cannot be characterized by IC50 value. These inhibitors increase Km and decrease Vmax. Example is diisopropyl fluorophosphate is an irreversible protease inhibitor. These are some of the therapeutic uses of irreversible inhibitors given in the table. Let us see what is suicide inhibition. It is another type of irreversible inhibition. In this case, the target enzyme converts the inhibitor compound into a reactive form in its active site. They are also known as mechanism based inhibitors or transition state analogues. Example, DFMO. Another example is allopurinol. It is a suicide inhibitor of xanthine oxidase. Allosteric inhibition. Allosteric inhibition is a type of enzyme regulation in which inhibitor binds to a site other than the enzyme active site. This additional site to which effector binds is termed as allosteric site. When these effectors bind to the protein, results with a conformational change and causes enhancement in activity is known as allosteric activators. When they decrease the protein's activity, they are called as allosteric inhibitors. Let us see what is the mechanism of allosteric regulation. The allosteric effects or mechanism is well explained by the concerted HWC model put forth by Monod, Wyman and Changeox. Another model which possibly explains the allosteric regulation is sequential model given by Koshland. Both the models postulate that enzyme subunits exist in 1 to 2 conformations, either tensed or relaxed state. Let us see the concerted model. This model is known as symmetry or MWC model. It postulates that enzyme subunits exist in same conformation. They are connected and a slight conformational change in one subunit is necessary conferred to all other subunits. In the absence of any ligand or substrate, the equilibrium favors towards either of the conformational states. R state has higher affinity than T state. The most successful application of this model is this regulation of hemoglobin. Let us see what is sequential model. In contrary to concert model, sequential model states that subunits are not connected so that conformational change in one induces a similar change in the others. When a subunit randomly collides with substrate, an induced fit converts a subunit from T state to R state. Let us see morphine model. The third model is a dissociative concerted model known as morphine model. This is physiologically significant homo-oligomeric tetramer structure. Oligomer dissociation, conformational change in dissociated state and reassembly of oligomers assist the transition in morphine model. There are different types of allosteric regulations. Homotropic regulation, it is a positive modulation. The modulator is a substrate as well as a regulatory molecule of the target enzyme. Example, carbon dioxide is a heterotropic modulator of hemoglobin. What are the importance of enzyme inhibition? Enzyme inhibition is important for understanding the regulation of enzyme activity within the living cells. It is useful in elucidating the cellular metabolic pathways by causing accumulation of intermediates. It is also useful to identify the catalytic or functional groups at the active site of enzyme. It provides information about substrate specificity of the enzyme. It is also used to study the mechanism of catalytic activity. To summarize, in this module, we have learned different types of inhibitions and its application in various fields. Thank you.